Thanks to our incredible first round of speakers. I'm Amy Elliott from Form Bio. I run our product shop, and I'm going to talk to you a little bit about the black box AI and some of the squiggly lines that we heard about from some of the previous speakers, and what we're doing to try and help this incredible community move things forward to become life-changing therapeutics. So a couple things that I really want to focus on that you've heard a lot about from others are overall themes of the importance of metrics, how we measure things, how and when we evaluate them. So three of the really important pieces for a successful gene therapy are safety, efficacy, and manufacturability. Manufacturability gets a lot of attention right now because that's where we have a lot of incredible challenges as a field to figure out how to scale up, how to be reproducible, how to do that in a timely manner, and how to do it at a cost that's actually gonna do the job. Efficacy is another really important one. It's got to work. It doesn't matter if we can get it there. It doesn't matter if we can get it there with the right dose if it doesn't actually do the job. And so that's a critical piece of the puzzle. And then safety is always a concern. We heard a lot about the amazing history of these viral technologies and how that has changed a lot over time to try and overcome some of the early issues, but safety is still an ongoing concern. So in the more than 3,000 gene therapies that are currently in development, these are three of the main issues that we need to address, and that's said by many people. So a couple things I want to talk through. I want to take a little bit of a step back first and look at the industry and what some of our, so we can celebrate some of our successes and talk about some of the challenges. So I've got three examples here where we have gotten through successful FDA approval. And I want to talk a little bit about how this has gone down. So this first one, and most of you are going to know what these are, right? So the Novartis one, they acquired the asset at phase 3. Hey, it met its primary and secondary endpoints. That's great. It was about 10 years and $1 billion in investment to get it there. So we're still facing incredible timelines and incredible cost. Similar story for Roche, except it was developed through phase 1 in an academic environment. The way that these are developed and licensed out from discovery through to early clinical trials and then into late stage is an incredible series of handoffs. And so the other thing that I want to point out is that everybody has different criteria for determining what a good profile looks like, and where these handoffs happen make a difference in how we are evaluating them. Same thing for the third one, met primary and secondary endpoints, acquired late stage, so presumably quite de-risked, right? Still an incredible amount of money and time for investment. However, the success of the gene therapies are incredibly life-changing, which you got to see from some of the amazing videos that Jude showed this morning. It's a worthy process. I want to call these design opportunities, some of the challenges that we're having. Because another theme that you're hearing from some of the earlier talks is the faster that you can get to a design that's going to work, that's going to be successful across our three pillars of safety, efficacy, and manufacturability, the more likely you are to have a clinical success. And hopefully, right, the faster that you'll be able to get there. So I don't want to dwell on the things that are problematic, but I do want to take a moment to acknowledge that, for example, the Pfizer asset was primarily characterized by TEM. So we heard a lot of the great success of characterizing with NGS data. Well, what you get with TEM is a really excellent picture of the full particles versus the empty particles. You have no idea what those full particles are full of. So this particular one had 88% full vectors, which under normal circumstances would be an incredibly great, let's go forward success. It didn't meet any of its endpoints, so it utterly failed at efficacy. And that's partially probably because it wasn't well characterized in terms of what are the genomes that we're actually delivering. You actually have to make that protein, right? The Astellas one, I wanted to bring up a little bit because that was an incredibly high dose, the highest dose delivered to date. So the other thing that's important about how you do the formulation and the design up front is so that you can keep the dose as low as possible so that we're not delivering a bunch of virus. The Unicure one I have up here is an interesting one. I think of this as a competitive redesign. So they were watching the market, and a better coding sequence was published for factor 9 with the Padua, right? So they acquired that better CDS, they redesigned, they did an additional 5 years of clinical investment, and they got it through to FDA approval. So it doesn't have to be a deal breaker if you have a design failure. you have the opportunity to redesign. But if you don't do the correct characterization all the way through, then you are spending a lot of money and you're missing a lot of opportunities. And so at FormBio, this is the thing that we focus on heavily, is where and when to do what kind of characterization for your drug substance, and how do you optimize that to achieve the best possible outcomes. So I'm not gonna walk through all of this stuff in real detail because I want to keep everything moving along, but I think one of the things that we're focusing on heavily, and you got to see a great idea of this from Brooke as well, is we really need to get better as a field at defining the metrics for design success. We have to design our metrics early, and I think often, right? I think of this like the old days with Microsoft Word. You hit Control-Save early and often, right? So that you don't lose anything. It's the same thing here, right? You have to think about, for your individual indication, which of these things are going to be the most challenging and the most important, because it's going to be different for everyone. You have to establish what those metrics are going to look like, because the FDA is not going to tell you because they don't know, which is difficult, right? Because it gives us the freedom to define the assays that we're going to use. It gives us the freedom to define the metrics, but it also means that it's the Wild West, and we don't share information as easily across industry as you do sometimes in academia. And so a lot of things that are being learned in one organization are not being learned elsewhere. So I think this is something that is a conversation that we need to continue having. Examples. So I think from a technology perspective, we're at a really incredible time. I think one of the things that Jude's talk really brought up for me, because I've been in molecular biology and structural biology for a very long time, is how the technology has changed to give us different kinds of resolution and different sizes of data. So what we can do today with the omics data, it doesn't matter if it's proteomics, genomics, it's all large data. So in order for an AI or a machine learning or a deep learning technology to work, there has to be enough data for patterns to be recognizable. And so this was just not really a viable option when you're looking at qPCR values and you've got one per sample, right? It'll take 150 years to get enough data to do anything with a pattern matching perspective. So I think one of the things that's really important right now is this intersection of the development of the large language and other types of deep learning models with the availability of the data to train them with, and then also advanced characterization, which is a lot of what you heard about already with the different types of features that we're looking for. How do we couple the features that we can identify in the next generation sequencing, really third or fourth generation sequencing now, data with actual outcomes, so that we can design for the outcomes we want, and we can design away from the outcomes that are going to be problematic. So this is just something that I wanted to show. It's the other thing about you're welcome. One of the other things that's really important about the data is when you're training a machine learning model, these things are best at whatever specific task you can give them to do. Almost none of them generalize well. If anybody is telling you that they're lying, don't believe them. But one of the things that's really happened is from 2017 forward, you can see on this y-axis, it's 10 to the 5, 10 to the 10, 10 to the 15. So this is the size of the data sets that are becoming available for like proteomics type data. So these are the data sets that are being used for your alpha folds and all of the amino acid side of the house type things. There is a hell of a lot of data there, right? That's incredible. This is a picture of our data set. I'm sorry I'm blocking this one, but what I want you to see is the trend here. So we're starting, these are the number of reads from the NGS data. So this is 5E7, 1E8, 1.5e to the 8. And I just want you to appreciate that. We have a similar trajectory of massive increase in the data that we're getting, but it's the right data. So you have to train the models on the data that's actually going to be relevant for what you want to do. And that's something that we've really focused on at Form. Last thing that I want to show is, so this graph is full genomes on the y-axis and partial genomes on the x-axis. This is from long-read sequencing data. Each one of these dots is a data set that's coming from our client base. So these are where our clients are coming in with their designs. So in the top left, what you really want to have is a lot of fulls and a small number of partials. This is not even looking at contaminants. But what I want to show here really is the incredible spread that we see when we're coming in at the beginning or even in a later stage program when you're trying to characterize your drug. It's all over the place. It's all over the place largely, and I think you'll probably hear about this from Fred a little bit, because the protocols are also all over the place. So you have to standardize your process. You want to design for your process while you're designing the drug substance so that those things are reproducible and they end up with outcomes that are going to fall in that green space. The great thing about where AIML fits in here is the iteration to get from way down there to way up here is much faster, and it saves a lot of money and lab time. So this is my homage to the black box. It's actually a black box, sorry. So the optimization engine, I'm not gonna walk you through actually the in-depth detail. They're happy to talk about some of that later. But the thing that I do wanna point out is in addition to having the right data, it's important to have the right objectives and constraints. So for our purposes, right, we take a nucleic acid-based perspective on what it is that we're designing, as opposed to an amino acid base like you would do for an antibody or a biologic. So we need to do things like percent GC content is maybe gonna matter, the codon adaptation index. We have different things that we can use to address the trade-offs that are gonna happen in vector design. Because if you need to overexpress something, the typical way to do that with an off-the-shelf codon optimizer is just to crank up that codon adaptation index, right? So that you have as many permissive tRNAs as possible. Well, what do you lose if you crank up the expression, right? Maybe you introduce a bunch of secondary structures that are gonna be problematic for replication, so you get a bunch of truncations, et cetera. There's never a free lunch in biology, right? That just never happens. So understanding the trade-offs is something else that we can do both from a design perspective and from an evaluation perspective. Similar thing with constraints. So what we do is we'll create a vector design space that has all of those constraints accommodated. If there are a bunch of promoters we can try, like you saw from Adam's talk, we can do a bunch of different design things. And then we've got two things that we do as a company. One of them is rational design, and the other one is optimization. The rational design part is a big place where NGS data comes in, because we can take a look at how those different elements are operating, the promoters, the enhancers, whatever, in the genomic context they're in. And then we can collect a diversity of data that helps the model understand what the patterns are that are going to work and what the ones are that won't work. And then our optimization engine, black box. But at the end of the day, what we get out of that, putting everything ahead, so we're even doing the design up front for how we're gonna do the optimization. It's really critical, and that gets us to a better optimized output. White boxes, but still too much detail, I think, for today. Feel free to ask questions later. So the places where Form really tries to operate are up front in the design and synthesis part of the equation, and then after viral production and you're getting into your drug substance characterization. So we really would like the industry to spend their design time up front and the optimization time up front, so that when you get to the characterization part, you've saved a lot of time and a lot of money, and you have more room to move. If you end up getting all the way into phase 1 and you find out that your promoter's a disaster, there's a lot more comparability studies that you have to do to try to go forward, and that isn't well defined by regulatory in the same way that it is in some of our, let's call them sibling industries. So it becomes really much harder to make changes the later you are in the game. Similarly, I mentioned earlier, I think it's really important to think about design for your construct itself and also for your process. And this is a place where that next generation sequencing data, like Brooke laid out really nicely, fits into both the design validation and the process validation. You can use that data in multiple places to make sure that you're doing the right metrics for the right outcomes. So lastly, I just want to walk through a couple of slides showing some of the things that we're seeing. This is some of this you've already seen. So when we do our characterization with the genome integrity analysis, we can do things like identify all the contaminants. This is the rep cap, for example. We can do that by percent and by count and the host genome. We can do things now like actually we're translating all of the CDS competent containing fragments, so we can look at all those partial genomes and see if they're long enough to actually make the protein. And this is something that you'll see with some of these partial genomes that still do the job, right? If you get the protein in there, it still does the job. So how much of your full is actually competent to be therapeutic is a question that this data allows us to assess. And then lastly, manufacturability. This is just showing the mutation profile. So this is, let's see, 1%. So we can look at the distribution of what all of those contaminants look like, and we can say, like, well, 1% is pretty darn good, right? All right, in terms of our design and redesign, I'm calling this design and redesign because it doesn't matter where we're starting. Our technology is agnostic to where you are in the process. We would like to do it earlier so that we have more flexibility and so that you have more time to make changes and to get to the right one. But we can do this work at any time. So a couple outcomes that I want to show. This is a data set where we're looking at full genomes on the y-axis, and then in blue is our client's starting vector, and in black is the FormBio optimized vector. And so I'm using the full genomes here as one of the possible safety metrics. Again, like NGS sequencing does not tell you anything about the capsid, so full disclosure. But we are seeing pretty routinely a significant increase in full genomes. Thinking about efficacy, when we're looking at expression, this is copy number over GAP-DH on the y-axis. Again, we're able to pretty commonly significantly increase expression. I've just got new data that on this exact same construct, if I tune the optimization differently, I can reduce it because sometimes overexpression is toxic, and this is a case where overexpression is toxic. So we're figuring out how to reduce that. And lastly, on manufacturability, this is looking at titer in vector genomes per mil on the y-axis. And the thing that I want to show here, in blue are the original constructs, and in pink are the ones that we optimized. And so this is not significant yet, and this is a crazy outlier, right? That's an incredible 400 and something % increase in titer. But what we are seeing is consistent improvement in titer with all of our optimizations. So all that to say, our 3 pillars that we're focusing on as a company to try to help improve, we are seeing significant improvements in. So the last thing I wanted to do was zoom out, I guess, and talk a little bit about where AI and in silico tools sit in the future of AAV. So there's what we're doing, but there's a great big landscape of AI tools outside of that. And there are lots of places where I think this is going to be really helpful and important for us as a field. So these are listed here. I think a couple of the ones that I want to point out that you've heard some about today are complex data integration and interpretation. Having the right data is not useful to you if you can't interpret it, right? So there's a whole extra layer that's going to be really important there, and AI is getting pretty zippy at helping you summarize and interpret data. And then the other thing that I wanted to point out is data generation is increasingly cost effective. So as the cost of the sequencing technology comes down, the more available that's going to be to us all to be able to use. This is a graph that shows kind of where along the discovery to clinical development, manufacturing, commercialization, there has been a massive surge of technology companies. I've got data management along the bottom because that's a little bit different. But I think the thing that I want to point out is that for vector design and vector optimization, we've got a whole lot of companies that are wading in, that are focusing on different aspects of the problems that we're trying to address. So there are a lot of really good opportunities to take a hold of this technology as long as you have the right data, and I hope help move the field forward. Okay, I want to give a shout out to our Genome Integrity AAV working group. If you happen to be in the audience, if you would raise your hand, if you don't mind. Yay, thank you all so much. This has been an incredible collaborative effort. We love this group so much because it's a place where this community can come together and talk about things like standards, try to move the field forward as a group where we're sharing our successes and we're sharing our failures and we're learning what works and what doesn't so that we can grow as a field. This period, I guess, last year we did the bioinformatics workflow. This year we're doing the library prep. So you've heard a lot about that today. The library prep is really challenging. It's the wild, wild west. So we're working as a group to try and figure out, how do we create standards that are testable for that so that we can help everybody standardize. And with that, I will acknowledge our collaborators with Resilience and Solid and UT Southwestern and Azenta, and I've got our AAV working group up here as well.
All right, thank you, Amy.